initiate this lab, we created a series of dilutions from a stock solution. Then we transferred each dilution into a separate cuvette. We then moved on to a spectrophotometer to begin the next portion of our lab. Before working with the spectrophotometer, we made sure to understand Beer-Lambert's law, which includes absorption, path length, and concentration. Absorption is found by using scans, and concentration can either be calculated using known information about the substance or by determining the concentration based on absorbance. Path length is the distance of light that travels through the sample, which is usually 1. Absorption and concentration are directly proportional, so as absorption increases, concentration increases. If you fail to zero the machine using a blank, your results will be off because the machine will scan more than just the substance you are trying to analyze. First, we placed a blank in the spectrophotometer in order to have it zeroed before scanning our series of dilutions. Before placing each cuvette in the spectrophotometer, we wiped 2 sides of the cuvette. Once we put the cuvette in the spectrophotometer, we made sure to place the sides that were just cleaned in the beam and to close the spectrophotometer before scanning. We then scanned our strongest dilution to get our wavelength, and also scanned each dilution to get their absorbances. It is important to scan across all wavelengths when using the spectrophotometer, because doing so helps you to determine the wavelength of maximum absorption, also known as lambda max. Scanning across all wavelengths also helps you to differentiate the actual lambda max from any interference that the plastic cuvette might cause, allowing you to obtain the most accurate values and data from the spectrophotometer. Lambda max is the term that refers to the wavelength along the absorption spectrum where a substance has its strongest photon absorption. We recorded the absorbance of each of our dilutions in our lab notebook and carefully removed the cuvettes from the spectrophotometer. Lastly, we cleaned our area of work and made sure everything was left as it was found.